Okay, so we'll start today's session. Let me share my screen. Okay, so to begin with, we will look at few questions, like previous year assignment questions. But before that, I will just briefly show you. Like suppose we discussed about you will do chromatographic techniques for purifying a protein and all. And all these things depend on what is the isoelectric point of a protein. Okay. So how will you really know the isoelectric point of a protein? Okay. So suppose you are working for the very first time with a protein. So usually there are certain online tools wherein or online sites wherein you copy paste your sequence, amino acid sequence, and then you can get a theoretical pi value, and you can also see like different kinds like absorption coefficient of that particular protein of interest, etc. Okay, so there is something called, but before that there is uniprot database. This is a database wherein you just directly, like suppose like if we are talking about insulin. Okay, now you just search for insulin and you will see whatever, like whether it's from human, cattle, chick, okay, or pig or rat, mouse. You can just select on that. Suppose we are interested in human insulin. Okay, you just go there. So here what you can say is you have all the like organism number of amino acids that are there. Okay, you can directly go to the, like here, from by clicking here in these areas, like in these options, you can directly go to the, like first thing is like function. You can directly go to the sequence. Okay, or you can go to the sequence from here as well. Directly you can get the amino acid sequence from here. Okay. And then you can view the isoforms, different kinds of isoforms. You can also see the structure of insulin. Okay, so you can just rotate it here and there using your mouse or mouse pad. Okay, and see the chains. Hmm? So this is, and then you can click on these regions also to know what, which is the N terminal, which is what residue, like that. Okay, so these are few of the things. And so suppose, yeah, now right now we are interested in getting the isoelectric point of the protein of our interest, suppose. So we just chose insulin from here. Now this is the sequence. What you do is you just copy the sequence from here. Okay, and there's another called expassy-proteparum. Okay, so here what you do is, see here it's written that this is used for computing the different physical and chemical parameters of a given protein. Okay, so what you, either you directly paste the uniprot ID. So this is the uniprot ID here. Okay, this is the one. You can directly copy paste it, or because we have the amino acid sequence, you just copy paste the amino acid sequence and click on compute parameters. Okay. Now see here, you will have all the amino acid composition. It will give an approximate like extinction coefficient. It will also give an instability index. Okay, so this is how uh, you will have all these like estimated half-lives. Now you will have extinction coefficient assuming all cysteine residues are reduced, like there's no disulfide linkages, or assuming uh, there is disulfide linkages. Based on that, it will compute these parameters. So you can have a look at this. Number of amino acids, it will give the molecular weight as well, depending on the amino acid sequence. It will also give this isoelectric point here, okay, which is 5.22 for insulin. Now, based on this, whether you want to go for like suppose for purifying insulin, any suppose you have cloned the thing, now you want to overexpress and purify insulin protein. Now, based on this theoretical PI, you can choose which kind of chromatographic technique you want to go for. Okay, because you have to play around with the pH of the protein, because a certain pH higher than this PI or lower than this PI will give a characteristic charge, which will eventually be used for suppose ion exchange chromatography, or if it has any other like imidazole containing group, like histidine containing group, or there are several histidine residues that has been added or cloned into the recombinant protein. Okay, so then you can go for affinity chromatography like that. Okay, so this is one of the things I wanted to like briefly show you. Okay, now let's start, like just look at few of the previous assignment questions. Okay, now the first mammalian clone, Dolly, which was a sheep actually. So with it was cloned in the like which time period? Does anyone know about it? Like, has anyone heard about Dolly, the clone, the first mammalian clone done, and which type of cell was? Yes, it's 20th century. I think it's around 1990s, in that period. It was done, I think 1996 or 97 something. So it will be option C. It should be. Yes. In 1997, what happened was Wilmot and his colleagues in Edinburgh, they cloned a sheep named Dolly. And this was the first cloned mammal. Okay. What they did actually, they had this particular sheep. What they did, they took the egg from this particular sheep, like this was the donor, and they took the egg, removed the nucleus, and this enucleated egg was fused with a somatic cell from another sheep, okay, having a different kind of phenotype, now. So yeah, they transplanted a nucleus from a mammary gland of a fin Dorset sheep. This was the breed of the sheep, into enucleated, this one, into enucleated sheep of a Scottish blackface ewe. Okay, ewe is a female sheep. The nucleus egg combination was stimulated using electricity to fuse the two and stimulate cell division. And the new cell, hence which was resulted, this particular cell divided and was placed in the uterus of a blackface ewe to develop. Now this fused one was introduced inside the uterus of the same species or the same breed of sheep, and then developed. But then the offspring that was obtained like did not have the characteristics of this particular like in the organism or the black faced sheep. The phenotypic characters were very different, rather it was similar to the one from whose cell it was used. Okay, so the dolly was shown to be genetically identical to the fin Dorset mammary cells and not to the black face ewe, which clearly demonstrated that she was a successful clone. and but it actually took 276 attempts before the experiment was successful. Okay, so in general, the question what it wanted to know, like it was around 1997, so the answer will be 20th century. Okay, now this we had gone through earlier also. So prokaryotic ribosomes are composed of larger dash and smaller dash subunits. So what will be the answer? Yes, option A, 50 and 30th subunits. And what about eukaryotic ribosomes? 60s and yes, 60 and 40. Okay. So, but then when these are like separate subunits, right? So in case of prokaryotes, when 50s and 30s joins, what is the combined ribosomal ribosome that is formed in prokaryotes? Yes. And in eukaryotes, can you repeat? Yes. Yes. So, so why is it so? Like, why isn't it like directly additive? Like 50, 30, would it like, shouldn't it give us like 80s? It is giving us 70s. Like, whatever you are aware of, you can just try and answer. It's completely fine. So, basically, the S here represents Swedberg unit, right? And it depends on the amount or the time taken by this particular macromolecular complex to like settle down, or time taken for it to move down during centrifugation. Okay. Now, so that isn't really the overall, like it, all this depends on the overall constituent of the macromolecular complex, or rather ribonucleoprotein complex, because ribosomes are made up of RNA, like ribosomal RNA and ribosomal proteins. Okay. Now, in case of suppose 50s, 30s, so in 50s you have 23s ribosomal RNA. Again, this 23s is like the rRNA, that is, I think it's around 2.8 or 2.8 to 3 kilobases long in case of 50s, and in case of 30s, it's around 1.5 kilobases long. So the ribosomal RNA. So then we and it's 16S ribosomal RNA in 30S. So again, these S unit, that is actually, like in very simple terms, it's basically the time required by such macromolecules to settle down during centrifugation, or rather ultracentrifugation. Okay. So the 30s and 50s subunits combine to form 70s ribosome, or in eukaryotes, 60s and 40s will combine to form 80s ribosomes due to differences in how the sedimentation coefficients work during ultracentrifugation. These values are not additive, but rather these depend on the shape, size, and density of the particles. Okay. So again, like that is what I was talking about. Like when we talk about um 30s subunit and 50s subunit. 50s is made up of 23s rRNA and 5s rRNA. 5s is like really small. It is around 120 nucleotide long, and 23s is rather huge, which is around 2.9 or 3 kilobases long. Okay, and this is like 16s rRNA for 30s subunit, which is made up of around 1.5 nucleotides. Like 1.5, which is composed of around 1500 ribonucleotides and 21 proteins, ribosomal proteins, whereas 50s ribosomes are made up of 31 ribosomal proteins. Now, when these are separate, uh, they have these compositions. But when these two combine together, there will be interaction between the two faces. There is inter-subunit surface, okay, where the large subunit and the small subunit combine together. Now, overall, this entity will have a different time or different rate or different density according to which it will like settle down during ultracentrifugation. So that's why it's not really additive like direct addition. Rather, this is depending on the sedimentation coefficient. Okay, so similarly for eukaryotic ribosomes, these are the different compositions. Okay. Now, the gram-negative bacterial cell walls are not able to retain which of the following? Can anyone try? Um, the answer, actually, we are talking about this. Yes, yes. So, yes, yes, yes. Please go ahead. So, like we are talking about the stain here, okay? Or the this one. So the answer will be crystal violet, okay? So, but before going there, we will look at what, because again, this retention, when what we are talking about the retention, it depends on the property of the, like mainly the composition of the cell wall in gram positive and gram negative bacteria. So here, so in case of gram positive bacteria, this is your like cell membrane having phospholipids and proteins. Then there's this peptidoglycan layer. Okay. But in case of gram negative bacteria, this is the cell membrane. Then there's a thin peptidoglycan layer. But in case of gram positive bacteria, the peptidoglycan layer is thicker. So this is a thin peptidoglycan layer. Then there's outer membrane, which is made up of like lipopolysaccharides. Okay. So due to this particular composition, gram negative bacteria are not able to retain certain kind of stains. So in case of gram staining, what happens? You suppose there are two types of stain. You apply first this particular stain or dye, crystal violet, and you actually apply iodine, which is a mordant, and then you use ethanol or alcohol to wash it off to decolorize. Now the ones which will retain the stain, like which won't be able to retain the stain, it will be washed off the stain like this dye will be washed off. And then if you add a counter stain, then the cells from where this particular purple dye has been washed off will be stained pinkish. Okay. Using the counter stain usually used is safranin. So what happens here is, in case of both gram positive and gram negative bacteria, both the cells affix the dye. Okay. And the dye complex is trapped in the wall because there is the thick peptidoglycan layer in case of gram positive cells. But uh, like in case of like dye is affixed, but when iodine is added, there is no such effect. So what happens is these crystals remain in the cell wall in case of gram positive bacteria because of the thick peptidoglycan layer. Whereas in case of gram-negative bacteria, the outer membrane gets weakened and the cell wall loses the dye because it has thin peptidoglycan layer followed by an outer layer of lipopolysaccharides. Okay, and then when you add a counter stain, this will like red dye masked by violet dye because being more darker in color, they will appear purple only. But here we can easily see the safranin color of the safranin dye in case of gram negative cells. Okay. So now the answer will be crystal violet. Okay. Now, so which of the following is the cofactor of the enzyme hexokinase? So what will be the answer? Okay. Magnesium ions. So hexokinase is which, like, do you remember in which step or in which metabolic pathway was it mentioned, or does it function? Yes. Okay. So let us just very briefly look at what happens in there. So we read that in glycolytic pathway, one is the preparatory phase where it requires energy or ATP, and another is the payoff phase wherein we get ATP molecules or ATP molecules are released. Okay. So in the preparatory phase, the very first step is catalyzed by hexokinase enzyme. Like in general, for when we talk about kinase enzyme, that is an enzyme that adds phosphate group to a particular molecule. Okay. So this is what glucose is converted to glucose 6 phosphate via hexokinase, and glucose 6 phosphate isomerizes to fructose 6 phosphate using phosphohexose isomerase, and then this is changed to like another phosphate group is getting added, thereby forming 1,6 bisphosphate. So another kinase will be playing a role here, and then this is broken down into three carbon compounds. One is glyceraldehyde 3 phosphate and dihydroxyacetone phosphate or DHAP. Okay. So now here wherever phosphate group is getting added, so that phosphate is being taken from ATP molecules. So in two of the steps, ATP molecules are being used here. Okay. But now when we move to the payoff phase, like using the three carbon compound, now this is getting converted to glyceraldehyde 3 phosphate, suppose. This is getting converted to like 1,3-bisphosphoglycerate, finally to phosphoenol pyruvate and pyruvate, thereby releasing ATP molecules. Okay. So this is very briefly the glycolytic pathway that we had. It's the same slide from the previous session. Okay. Now, so what, like you have to select the incorrect pair among the following. Okay. Yes, yes. Yes, yes, please go ahead. Negative marking, I probably not, because I'm not aware of the rules as of now, but or maybe there is, I think for 4 questions there might be. I have to check the rules and regulations. I'm not aware about it. So maybe you can write it to like the people who are conducting the examination. So I think there will be instructions as well in the question paper. So there might be probably. Okay. But maybe how it happens, like for 4 questions, 1 mark, like that, probably. I'm not very sure. Okay. Okay. So yeah. So out of these chromatographic techniques, basically the principle upon which these chromatographic techniques are performed. So which of the following is an incorrect pair? Yes. So what does it actually depend upon? Okay. And how is the charge rendered to the molecule or protein rather? Protein nucleic acids. Yes. And so, and what about the matrix, like to which the molecules are bound? Yes, yes, yes. Okay. And also, so when we talk about, like in case of anion exchange chromatography, the matrix used is an anion exchanger, or it is positively charged. Okay. So this way, sometimes the questions can be asked. Okay. So yeah, the answer will be, in this case, it is ion exchange chromatography, molecular mass. Now, in a PCR reaction, the denaturation of the double-stranded DNA is done by heating the reaction mixture at, what will be the answer? Uh, of, yes, 95 degrees Celsius. Okay. So usually it's 95 degrees Celsius because 110 degrees Celsius might be too uh, like too improper for the molecules to really bear that high amount of heat. But then 95 is also high. But in general, for denaturation, 95 is good enough to really separate the two strands. And then you can play around basically with the annealing temperatures and then extension timing and all those things. Okay. So, what are the basic steps in PCR reaction, in a PCR cycle rather? Like in yes, and then there is amplification. Okay. Okay. So, in which part, like the melting temperature, all these comes into picture? Um, and what about annealing temperatures? Yes. Again, it also depends. So, what does it actually depend upon? It can be little higher than that also, but again, what does it depend upon? Yeah, yeah, you are correct. It's right only. Uh, so it basically depends upon the primers, right? That will be used to uh, like extend the add on to the like basically complement like complementary nucleotides will be complementary bases or nucleotides will be added. Okay. So the primers have to first anneal or anneal to the template strand. Okay. So that annealing temperature is based on like different, we read about like whether what is the length of the primer, like as in the number of nucleotides in the primer, and what are the composition of the, or the number of GC base pairs or GC bases in the primer, because if there are higher content of GC bases, then the it should be optimum enough. Like the annealing should be optimum enough that it shouldn't be too tight, because in case of GC bases you have three hydrogen bondings, right? That is tighter. So it will the primers have to move away as well. Okay. So that particular thing should be taken care of. It shouldn't be too tight that the primer isn't moving at all. So the amplification would be improper. Or in general, the extension and amplification would be improper. Okay. So yeah, for this particular question, it is 95 degrees Celsius. So let's like very briefly look at just just go through the thing once. Okay, so there is this initial denaturing, which leads to melting of double-stranded DNA into single strands, and denaturation initiates the this particular cascade. This is the cycle. So you then lower down the temperatures to allow annealing of primers. Again, this particular temperature is decided based upon what is the composition, what is the the composition of the nucleotides of the primers, and what is the length, etc., and whether the primers are making any, clamp structure or any kind of secondary structures. Okay. Then there's elongation, and this elongation is usually done using thermostable DNA polymerase. Okay, and then and basically this is denaturation, which is more than 90 degrees Celsius. Then annealing, wherein you add primers, lower down the temperatures, add primers, and then you extend the strand, and then amplification. By repeating these three core steps, you basically amplify and get DNA strands in exponential numbers. Okay. So yeah, just briefly we'll go through the things. Primer length, which is usually 18 to 24 bases long, which is the ideal length. And this is long enough for adequate specificity and short enough for primers to bind easily to the template at the annealing temperature. Okay. When we talk about primer melting temperature, primers with melting temperature in the range of 52 to 58 degrees Celsius generally gives the best results. But again, see, this is the usual thing. But again, based on what kind of, if you're working with a gene that is kind of tricky or having, a larger number of secondary structures, the primer especially, the base nucleotide composition is such that it will give you clamps or secondary structures, or there are multiple amount of GC bases in there, then it can vary. Okay. So the GC content of the sequence gives a fair indication of the primer melting temperature, and it should be prepared in such a way that there should be a difference not more than 2 degrees Celsius. Actually, it can range between 2 to 5 degrees Celsius. Otherwise, it will result in poor annealing efficiency. Okay. Then primer annealing temperature. If it's too high, it will produce insufficient primer template hybridization. Thereby, the efficiency of the PCR will be lower, or yield of the PCR will be low. Or if it's too low, the annealing temperature is not really like up to the optimum range, then it will lead to non-specific PCR products. Okay, because it will bind to any other place, any other region, and your specific gene product or nucleotide sequence won't be amplified. Okay. Then GC content, the number of uh, Gs and Cs in the primer should be nearly 40 to 60%. And GC clamp, because GC forms a stronger bond as compared to A and D, the number of GC content at the 3' end of the primer should not be more than 3, or it will result in non-specific tight binding. Okay, wherever GC content of G and C are higher. Okay. And also, we should take care about formation of primer dimer. Okay, like there shouldn't be complementarity between the forward primer and the reverse primer. Okay, otherwise those two will be binding, and those two won't be really binding to the template strands, like their respective template strands. Now, a primer dimer is a structure forming a double strand-like structure which is formed by intermolecular interactions or complementarity between the two primers or primer sequences. If interaction is formed between two homologous or same sense primers, it is called a self-dimer, whereas if interaction is formed between two different primers, it is called a cross-dimer. Okay. Repeats and runs. Repeats are consecutive occurrence of dinucleotides, whereas runs are continuous stretch of a single nucleotide. Suppose CG, that is a repeat, CG repeat. But suppose A or G are repeated continuously, then that will be runs. Okay. So usually what is allowed is a maximum number of repeats and runs accepted is 4 dinucleotide repeats and 4 base pair runs, or continuity of base pairs. Okay. And then primer template homology. Primer should be designed in such a way that there should be no homology within the template other than the target site. Like it shouldn't be going on. This is to stop, or rather prevent non-specific binding of the primers. Okay. This will result in non-specific binding and amplification, and you will get a lower product, PCR product of your gene of interest or your sequence of interest. Okay. And this is like for a primer length of less than 14 nucleotides, this is how you should be calculating the melting temperature. And for a length of more than 13 to 14 nucleotides, this is how, like this is the usual formula to calculate the melting temperatures. Okay. Now, so we read about, okay, PCR amplification has been done. Now, how do you really visualize your PCR product? You run an agarose gel. Okay. So depending on the size of the your amplicon, or the nucleotide sequence or DNA sequence or RNA sequence, the percentage of the gel can vary, or it should rather vary, because so The size of the pores will allow different sizes of the nucleic acid to move through it. Okay. So this question, the optimum agarose gel percentage typically used to view an amplicon, or rather a sequence of around 30 kb long, is what should be the thing here, like what should be the percentage here? Like 30 kb is a very big nucleic acid size, okay, big macromolecule. Option. Actually 2% is also a quite high. Yes, option C. Because usually the thing is, for 2% or 3% or 1.5% gels, what usually happens is when it is like really low, like suppose 100 bases, only 100 bases long. And here we are talking about 30,000 bases. Okay. So to run such a large amplicon or such a large size of nucleic acid, this should be, percentage of agarose gel should be low, because if percentage of agarose gel is low, then the pores formed will be larger, and such a large amplicon will be able to move through the pores easily. Similarly, but suppose if I am using for only 100 nucleotides, I am using 0.5% gel, then that will just, there won't be any resolving as such. It will just easily move through such larger pores, and it can even come out of the gel as well. Okay, so for 100 bases or 50 bases or 90 bases long, suppose some amplicon is there. For that, we use like 2%, 1.5% gels. 3% will be again the more higher of like percentage of agarose gel if you are using, then the denser it will be, and the gel will be running slower. Okay. So like these things have to be kept in mind when you are practically doing the things. But for this particular question, for running such a large amplicon, the percentage of the gel will be 0.5%. Okay. Okay. Now, which of the following restriction enzyme cuts the DNA within its restriction site? Does anyone remember? We will be again summarizing the thing as well. Yes. 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 So it will be type 2. Okay. Now let us just go through the different kinds of restriction enzymes. Okay. So restriction enzymes vary in the structure, recognition site, or where they are exactly cutting or like digesting the nucleotide sequence, and what is the cofactor required. So type 1, what happens here is it cleaves at sites remote from its restriction site, and it requires both ATP and SAM, and it is a multifunctional protein. How? Because it shows restriction digestion as well as methylase activity. Okay, and there are three type subunits of type 1. One is having restriction digestion activity, R R form. Okay, then there's M, which helps in methyl transferase activity, like methylase, and then there's HSDS or S type, which is the which actually recognize the specific DNA site, binds to the DNA, and then performs its function. Okay, so which is, if we are talking about remote activity, like it is cleaving at site, it is recognizing a particular sequence, but it cleaves at around 4,000 to 7K nucleotides away from the recognition site. Okay. When we talk about type 2 restriction enzymes, it is the recognition site is usually 4 to 8 nucleotide long, and it cuts within that particular site. Okay. Now, due to this feature, type 2 restriction enzymes have application in genetic engineering for cloning purposes. And again, it has three subunits, M, R, and S type. Okay. This is the restriction. This is binding, DNA binding, and then this is methyl transferase. So the type 2 enzymes again are divided, okay, depending on the different features that these types of, like these type 2 restriction enzymes have. Like there's type 2B, type 2E, type 2F, G, M, S, T, like, and there are examples as well. this is from the previous class only, like previous session only, on like restriction digestion and types of restriction enzymes. Then there's type 3 restriction enzymes. The recognition site has two separate non-palindromic sequence arranged inversely oriented. Okay. Now, the cutting site is nearly not like type 1, it's like extremely remote, but uh, neither it is cutting in between the restriction site. The restriction activity is shown nearly to 20 to 30 bases or nucleotides away from the recognition site. And it has two subunits, res and mod. Mod is required for modification, methylation specifically of the host DNA, and res subunit cuts the unmethylated DNA. Okay. And just to recall what we mean by an enzyme unit. Okay. Uh, this is 1U when we talk about one enzyme unit. That is the enzyme required to catalyze the conversion of 1 micromole of substrate per minute under a specific set of conditions, specific set of experimental conditions, and which include the temperature, the pH, the substrate concentration, as well as the buffer composition as well. Okay. Which will ideally yield the maximal substrate conversion rate for that particular enzyme in question. Okay. And when we talk about the activity, this is usually expressed in milliliter. Okay, units, enzyme unit we talked about. This is micromole per minute, and enzyme activity is U or enzyme unit per milliliter. Okay. It relates to concentration and varies depending on the enzyme's role in metabolic pathways or any other like different kinds of metabolic pathways. Okay. And when we talk about the turnover number, it represents the maximum number of substrate molecules an enzyme's active site can convert to product per second when fully saturated with the substrate. Okay. And when we talk about star activity, which is like a non-specific activity shown by the enzymes. Okay. And when does it happen? So under non-standard reaction conditions, some restriction enzymes cut at sequences similar but not really identical to their exact or specific recognition sequence. So it again, this is due to certain buffer conditions. Maybe the glycerol concentration is higher. If some kind of enzyme has been incubated, if your DNA has been incubated with a certain restriction enzyme for too long, the incubation time at a particular temperature has been, suppose it's too high, or the incubation time is, suppose ideal thing is 15 to 30 minutes, and you have set the reaction or like put the reaction for around 2 hours, then there will be non-specific cleavages as well. Okay. So what happens here is it can lead to cleavage at non-canonical recognition sites or even complete loss of specificity. Okay. This is star activity of an enzyme. So we should like while performing your particular experiment or while setting a restriction enzyme reaction, it is like one should, because usually these are ordered from the companies and all, and they have a proper protocol or the defined time range or optimum time range, optimum temperature range where the enzyme is showing optimum function. Okay. If we like deviate from that particular thing, if we increase the incubation time too much or increase the temperature too much, it will lead to either star activity or lead to complete loss of specificity. Your reaction won't work. Okay. Okay. Now, alkaline phosphatase is used to, what will be the correct option out of these four options? Um, ligate insert and vector, yes, like it is one of it is used there, but don't you think like there are options which might fit better, like exact function of phosphatase? Like in general, when we talk about suppose any kind of enzyme, wherein this when we talk about phosphatase, it means it is removing phosphate. If we talk about nuclease, Yes. If we talk about nuclease, okay, or DNAs, RNAs, then it means it is digesting or cutting at the particular type of cutting at specific sites of the particular type of nucleic acid in question. So when we talk about alkaline phosphatase, this will be to actually remove phosphate groups like from a nucleic acid sequence. Simply, if we talk about the thing, so out of these four options, option D will be the correct one. Okay, definitely, this is one of the treatments that is used to ligate insert and vector. This is correct, but its exact activity, what is it is used for? This is actually to remove the free phosphate from the nucleic acid sequence. Okay, now, which of the following is not a criterion of a good vector? Yes. So, why do we really need any type of phenotypic change? Yes, but then y- yes, yes, like you are on the right track. So the main thing is if that particular insert has been or a particular gene sequence of interest has been inserted into the vector, then one needs to know, right, that okay, because not every particular sequence will be ligated to your vector, or you won't get like all the plasmids won't be recombinant. Okay, so to select out the recombinants from like the inserted, whether all the vectors have taken up your gene of interest, to really differentiate between those two, we are we need certain vectors which will give us certain type of phenotypic changes. Okay. So what can be the different phenotypic changes? Just some examples. So like we talked about antibiotic resistance, right? So if some gene has been inserted into a vector, and then suppose maybe we talked about insulin, right? We looked at the very first example there in insulin. Now you want to produce recombinant insulin. Now you inserted insulin encoding gene into a vector and you transformed into E. coli cells. Okay. Now you have transformed that recombinant vector into E. coli cells. But the thing is, not when you set the reaction, like you did all these cloning and everything, it isn't necessary that all the vectors or all the plasmids will have like the insulin gene inserted. Okay, insulin encoding gene inserted. So what will happen is, in order to select that out, the plasmid itself will have certain antibiotic resistance. So only when that particular antibiotic resistance encoding plasmid that has been, and also this is one case. Another case is not all cells will be taken up by the equal, not all plasmids will be, not all cells will take up all the plasmids that is there. Okay. So now whether the competent cells, the transformed cells have taken up your gene of interest, okay, then to select that out, the vector is having a certain antibiotic resistance gene, encoding gene. Okay. Now what will happen? You will provide, like this is one of the examples. Okay, you will provide suppose kanamycin or ampicillin is one of the antibiotics. Now ampicillin resistance, if that particular recombinant vector has gone inside the E. coli cell, then that particular cell will be resistant to ampicillin. Otherwise, ampicillin being an antibiotic, it will not allow or rather kill the E. coli cells, and it will not allow E. coli cells to grow. Only those cells that have taken in the vector, okay, the recombinant vector, then those cells will be able to amplify or rather grow on that particular medium wherein ampicillin is present. Okay. So one of the phenotypic change that we need is or that we use is antibiotic resistance. Then another phenotypic change, like we talked about reporter genes. Okay. Suppose fusing certain kind of fluorescent protein, like genes encoding fluorescent protein. Now, if that protein is getting there, then it will give a certain kind of glow. Okay. So it will give out, emit as fluorescence. Thereby, we can guess that, or we can uh, like confirm that, okay, this gene has been inserted there. Then we talked about blue-white screening, right? Like LAG-Z, whether if beta galactosidase enzyme is there, if it is getting produced. So then it will act on X-gal substrate, thereby producing colored compound. Okay. So these are different kinds of reporter assays, or even insertional inactivation, etc. Okay. So these are some of the phenotypic change that we really need to really distinguish whether the recombinant vectors have been transformed properly into the cells of our interest or not. Okay. And yeah, high copy number, low molecular weight. You need high copy number so that you get enough amount of recombinant vectors. And then you need low molecular weight because it will be easier to, it will, it won't really add on to the metabolic weight or metabolic function of the cell into which that recombinant vector is getting inserted. Because usually, suppose in case of E. coli cells, you are transforming this particular plasmid, recombinant plasmid, into E. coli cells. Now, that particular, in suppose insulin, E. coli cells don't need insulin, right? So, but you want insulin. So that is something that is foreign to the cells. Okay. So it shouldn't be a burden exactly to the cells, like too much of a burden. If it's a high molecular weight thing, even getting such a big insert or amplicon inside cells, that will itself be very cumbersome for the cells to take up. And then later, like replication, transcription and translation of that particular gene will be, will add on to the burden, metabolic burden of the cell. Okay. Now, uh, we talked about transformation and competency, right? So, which of the following compounds is used to prepare competent E. coli cells? Does anyone remember? Yes. Yes. So, what will calcium chloride do? Like any I, if you remember, you can tell or you can try out also. Like you can just guess or yes, yes, yes. We use calcium chloride. So what it basically does is calcium is a divalent cation. So it basically masks the overall charge that is there on the surface of the cell. Okay. Also, what it also does is because you are putting in the DNA, basically the plasmid DNA in the surrounding, and you are putting in the competent cells there. Okay. So that calcium chloride, if it is masking the charge on the thing, and DNA being negatively charged. Okay. So there is usually repulsion between the cell's outer surface and the DNA. But if the calcium chloride, calcium ions are actually masking the charge, it will help in the DNA also getting absorbed onto the cells. Also, because calcium ions and DNA being negatively charged, there will be attraction between the two and more amount of DNA. We can expect more amount of DNA to, or plasmid to get absorbed onto the cells. And then you provide, you talked about heat shock, right? So then you give heat shock so that you play around with the pore size of the cells, because there's lipid content in the cell membrane. So you actually play around with the movement of the lipids there, and pore, pores increase, pore size increase, and then whatever DNA has been absorbed, it can get inside the cells. Okay. So calcium chloride will be the correct option. Calcium chloride, and usually it is calcium chloride, or sometimes a mixture of magnesium ions as well, like magnesium ions as well as calcium ions are used. Manganese ions are also used. Okay. So depending on your need, but the usual like regular daily protocol for making competent cells require like calcium chloride and magnesium chloride. Okay. So yeah, what happens is the physical and chemical treatments. Physical, like the heat shock. Okay, the if we are talking about the regular routine protocol, and chemical treatments used during transformation, the temperature imbalances. Here we are talking about the heat shock. And because heat shock, not only heat shock, like we give heat shock at 42, then again put it on ice, which is around 0 to 4 degrees Celsius. Okay. So what it, and then you give CaCl2 treatment. It helps deal with the barrier to DNA uptake, such as charge repulsions and pore sizes. Okay. So calcium ion treatment and temperature imbalances are leading to uh, like like actually accelerating the process of DNA or plasmid DNA uptake. Okay. Now, spiroplast transformation is used to transform which of the following? Does anyone remember? Uh, can you repeat? Yes. So it is yeast. Okay. So what actually happens, like very briefly, you have your yeast culture cells, and yeast have cell walls. So that has to be removed partially, which produces spiroplast, and this partial digestion is caused by zymolase enzyme. Okay, and spiroplasts are fragile for osmotic shock, but these are competent enough because there are broken cell walls in between through those areas. Now you have a carrier molecule which can carry the DNA or plasmid inside the cells, and it is then taken up by the like it is incubated together at a particular temperature at for of like optimum duration, and then the those particular gene of interest or recombinant plasmid is taken up by the yeast cells. So the answer will be yeast. Okay. Now, this is again a vector with a restriction site located within URA3 was used to insert a gene of interest. After transformation into E. coli, the cells were plated on solid agar. This is the nutrient medium containing 5-fluoroorotic acid. But no colonies were observed after overnight incubation. So, which of the following inferences is incorrect? Does anyone remember? Like, this is part of those insertional inactivation, or how would you like really know that your gene of interest has gotten inside? We were talking about phenotypic selection, okay? So that is one like antibiotic resistance, some kind of insertional inactivation of a gene, reporter gene insert or reporter assays. Okay. So these are some of the things which will help us in knowing that okay, our gene of interest has gotten inside the particular cell. Okay. So does anyone remember? We will be going through it again. It's okay. So what happens here is, in general, when we talk about complementation of mutation, suppose we have a mutant, we have mutated a particular nucleotide sequence, and what happens here is a mutant host strain can be used to screen the plasmid containing the missing gene, and the transformant will grow only if the gene product from the clone will complement the function. Okay. So in general, genes taking part in metabolic pathway or biosynthetic pathway are routinely used for this purpose. So what happens here is what you really need is a host strain into which you are putting in your recombinant plasmid, and that host strain is usually deficient in a particular gene. Now, if the gene belongs to the biosynthetic pathway, the mutant host in this case are called as auxotroph, as host is dependent on the gene product or final product of the biosynthetic pathway as a supplement in media for growth. Okay. And so what will happen is it will be deficient the host strain into which the recombinant plasmid is getting inside. That is deficient in a like that won't be coding for a particular product, suppose. Okay. Now the and it will need that particular product to really grow in a particular set of medium. Okay. Now your growth media will be having the missing nutrient. Okay. Now the recombinant plasmid, if it is having our gene of interest, suppose that particular vector or recombinant plasmid will contain the gene to supply the gene product. And if that gene product is there, then the product from the recombinant vector or recombinant plasmid will complement the function, and it will help in completing the whole biosynthetic pathway in question. Okay. So what happens in yeast is, like, there are four different genes like HIS3, LU2, TRP1, URA3 as selectable markers. Now what actually happens here is, one is positive selection. So in positive selection, what happens is the host strain doesn't grow on the media lacking the functional gene, but if the host, it is a deficient host. Okay. Now if the host has taken up the recombinant plasmid, then the gene encoded by the recombinant plasmid will help, will supply the gene product required to grow in the media, and hence the host strain will actually grow on that particular medium. Okay. And negative, in case of negative selection, what is happening here is uh, a chemical compound, like some kind of nutrient is added to the medium, and then it is converted to a cytotoxic agent in the presence of a gene product. Okay. So if that particular gene product is present, it will actually not allow the cells to grow, or rather kill the cells. And as a result, it won't allow the growth of the wild type cells. But if the host strain has been transformed with the recombinant plasmid, and if it has the non-functional gene product and it grows in the, then what will happen is it will grow in the presence of the compound in the media. This is what is exploited here in case of URA3 gene, which is present in yeast. So it codes for this OMP decarboxylase. Okay. And this is an enzyme, 5-fluoroorotic acid is converted to fluorodeoxyuridine. Yes, yes, please. Yes. Oh, it's okay, okay, just, is it better now? Like, is it better? Hello? Okay, okay. So should I repeat from the negative selection? Okay. So in negative selection, what happens is some amount of nutrient or some amount of uh, compound or reagent has been added to the growth media. And if what happens is this particular reagent won't allow the wild type cells which doesn't have the recombinant plasmid, those won't be able to grow. Okay, those particular cells or host cells won't be able to grow. But if the host cells have taken up that particular recombinant gene or like recombinant plasmid, then this will have a non-functional gene product, because there is something has been inserted in between, and so the gene product will be non-functional, and hence, like, the cytotoxicity won't be there to the recombinant host cells, and then it will be the recombinant host cells will be able to grow in the presence of that particular uh, compound in the present in the growth medium. So what happens is URA3, this is a particular gene which encodes for OMP decarboxylase. Okay. In yeast, what happens is this codes for OMP decarboxylase, and an active enzyme processes this particular 5-fluoroorotic acid to toxic fluorodeoxyuridine. Okay. Now, if decarboxylase is there, then it will actually metabolize this to form this toxic agent, and then this won't allow the cells to, like, host cells to grow. But suppose this particular gene has been inserted in between in the URA3 gene in between at a site in between URA3 gene. So URA3 won't be able to function properly, right? So what will happen is it will form a non-functional decarboxylase. So if non-functional decarboxylase is there, then it won't really act on this particular compound which is present in the growth medium, and this toxin won't be produced, and hence the cells or the host cells will be able to grow. So if the cells which are able to grow, that has actually the insert in there. Otherwise, which are not able to grow, didn't actually get the insert in between. Okay. So this is actually one of the examples of insertional inactivation. Okay. Because due to this insertion of the gene of interest, suppose gene of interest or a foreign gene, what is happening is the function of the wild type gene, which is URA3 in this case, is getting inactivated. Okay. So yeah, so generation of this cytotoxic agent kills the cells carrying functional URA3 gene product. If there is insertion, it will inactivate this functional decarboxylase and help the host cells to grow. So, so in case of this question, it is asking the same thing. A vector with a restriction site located within the URA3, like in the diagram, if there was BAM H1 site, then a particular insert has been inserted into that particular restriction site. You have inserted a gene of interest. You have transformed the particular recombinant plasmid into E. coli cells, and the cells were plated on solid agar containing this particular compound. Okay, because this particular compound is acted upon by decarboxylase, OMP decarboxylase, encoded by URA3. Okay. However, no colonies were observed after overnight incubation. Okay. So if no colonies are observed, that means the gene hasn't been really inserted into URA3 wild type gene sequence. And yes, screen is okay. I will try sharing it again. Just, okay. So maybe like it's visible for everyone else. Okay. So sure, sure, sure. Sure, sure, sure. Yes, yes, sure, sure. So maybe I should continue. In case he's joining, just let me know. Okay. Okay. So if no colonies are observed, then that means URA3 is functional because it is coding for functional decarboxylase, OMP decarboxylase, which will eventually convert this particular acid compound, like orotic acid, into a toxin that will eventually kill the cells or won't allow these like E. coli cells to really grow. But if URA3 would have that particular gene insert, then it won't be able to code for a functional functional gene product. Then colonies would have been observed. So if no colonies are there, then which of the following inferences is incorrect? 5-fluoroorotic acid was converted to this toxic compound. Okay, like how we saw in the this one. Okay, this is the general case. If there are no colonies, then this particular compound which is there in the growth medium, that is acted upon by decarboxylase encoded by this particular wild type gene, and this toxin is produced, thereby not giving any colonies. Okay. And OMP decarboxylase was non-functional. if it would have been non-functional, then it would have allowed the colonies to grow. So this is the incorrect option. Okay. But if the gene of interest has not been inserted into the wild type gene, then that is why functional decarboxylase is produced, thereby uh, like forming this particular toxic product and not allowing the colonies to form. Okay, so option is option B out of these four, because we have been asked like which is the incorrect option out of these four. Okay, now this is like the SDS page ones which we were looking at. A protein is in the native state. Sorry, in its native state, it exists as a tetramer. So the molecular weight of the protein is found to be 48 kilodalton in denaturing conditions. Okay, so the native molecular weight is, if it's denaturing conditions, ideally, like it will be running at its monomeric form. Okay, and so if it is existing as a tetramer, so what will be the native molecular weight of the protein? Option C. Yes, like into 4. Okay, yeah, it will be 192 kilodalton. Now, when we were talking about electrophoresis, so the electrophoretic mobility of a protein is directly proportional to which of the following? Uh, do you remember? Then you can try. Charge. Yes. Yes. It is charge. It will be option B. Now, just to summarize, so electrophoresis is the migration and separation of charged particles, which is under an applied electric field. Okay, and there's anode and cathode, like two one positively charged electrode and negatively charged electrode, and separation impact on the ionic particles results from contrasts in their speed, which is the result of the molecule's portability and the field strength. Okay, portability or M of an ion not entirely set in tone by molecular size, shape, and charge, and the temperature during the separation. It is consistent under the characterized electrophoretic conditions. So, if this is your charged molecule that is moving, then there will be electrostatic force. There is the electric field that has been applied, and there will be a viscous drag as well, or frictional force on the opposite direction, which will affect its mobility or movement. Okay, or movement of the charged particle. Now, when we talk about like if we talk about the electrophoretic mobility, the term, it is directly proportional to the charge and inversely proportional to how viscous the medium is. Okay, and what is its radius, size, or radius and shape of the molecule? So, if we, because the question had asked about direct proportionality, it will be charge, and inverse proportionality will be viscosity, size, and shape. Because the higher, if the medium is too much viscous, if the size is too large, okay, so the movement will be retarded, right? Okay. Now, when we talked about SDS page electrophoresis, the pH of the resolving gel in SDS page is, yes. And what about the stacking gel? 6.8. Yes, 6.8. Okay. Now, the glowfish are a genetically modified fish that, what will be the correct option out of these? So, option, can you? A, yes. So, it will be expressing fluorescent protein. Now, um, when we talk about fluorescence and then chemiluminescence, so basically fluorescence is given off by fluorophore, and which is again, it will absorb a certain kind of light, and then due to the movement or transition of electrons across the different energy levels, this is given out. Whereas in chemiluminescence, like a uh, particular chemical reaction takes place, that gives the product hence formed, gives rise to the signal that is obtained. during chemiluminescence. Okay. So this is the difference. Like chemiluminescence is the emission of light or signal as a result of a chemical reaction. And in fluorescence, the light is emitted from a substance that has absorbed energy previously. Okay. Chemical reaction forms low energy products from high energy reactants, because that is how a chemical reaction in general proceeds. Whereas in fluorescence, what happens? Electrons are excited, definitely upon absorption of light or energy, excitation of a particular wavelength. Excitation of electrons due to absorption of external energy takes place. And after the completion of a reaction, you get to observe the signal in chemiluminescence. And as soon as you remove the source of energy, remove the source of the energy or the light or radiation, as soon as you remove that, you get fluorescent signal. Okay. But in case of chemiluminescence, until the reaction is complete, until that particular product is formed, you won't be getting the signal. Okay. Okay. Now, in the toxoid class of vaccines, like toxoid is basically some kind of toxic product, or the toxin that is administered. Okay. The dash is used for immunization of the subject. Subject means, suppose us, like mammals or humans, like rats, whatever, for vaccination. So in case of toxoid class of vaccines, the dash is used for immunization of the subject. Any idea? Option? Yes. Yes, inactivated toxic compounds. So, if we talk about, like, very briefly, the types of vaccines, one is like live attenuated vaccines. You basically inject the live organisms, but a little, like, it won't be really that much pathogenic. Another is like you inject the killed antigen, inactivated. These are the different types. Or you actually take in some kind of some part of that particular antigen, which is like antigen, and inject it, or like administer it into the host. Okay. And then there's toxic, like these toxins which are inactivated, which are administered inside into the subject. And there is again RNA based vaccines, like we have seen, like a spur in case of COVID-19. So, and also this is one of the sites which gives, like, very nicely a comprehensive view of what are the different vaccine types, and like very short but concise definitions. You can go through this, like what are the different kinds, if you need to just know more. But in case of, for this particular question, it will be inactivated toxic compounds. Okay. So, okay then, that's it for today. And all the best for your exams. The exam is on 26th, is it? 27th. Okay, okay. All the best. And like this discussion forum also, in case you have any question or anything, you can always write back. Okay? There. Okay. I think it will be objective type MCQ. Okay. Thank you so much for joining. Oh, okay, okay. That's all right. That's all right. The recording will be there. I will upload it in YouTube. So it's okay. You can see there. Oh, thank you so much.